11. This is also, more importantly, she is an expert in chemtrails, weather control, synthetic biology. She's the preeminent researcher in this of what's happening in your skies that affect you on a biological or synthetic level. This is very cutting-edge stuff. The dark agenda of synthetic biology. Please, I won't waste any more time. Welcome the very wonderful Sophia Smallstone.
Thank you. This actually is an hour or so, this presentation. We might not have time for questions, but you can ask me at the banquet, or you can come to my table tomorrow and ask me as many questions as you like, which I probably won't be able to answer. So the thing about what's going on now is, how big is the big picture? And I used to think that if you went outside the box, you could see more. And a lot of people believe that this settles at the level of power and control, that if they have all the power, they have all the control, that's what they want. There's actually something beyond that, and that's what I've started to explore. The big picture, I may be wrong, but I want you to follow the dots that I connect, which are basically in the form of a square. I'll start here, I'll go here, I'll go here, I'll go there, and a shape will be made. The terrain is huge. It's much, much bigger than I ever dreamed of. And so it's about really impressions and perspective. And I had a friend who's now no longer alive, Harriet. Harriet used to keep me awake at night on the phone and tell me things until 1 o'clock in the morning, notes that she had taken from listening to radio shows. She really taught me how to see. So, the real voyage of discovery consists not in seeking new landscapes, but in having new eyes. Now here is a photograph, and here is an impression. So I am going to give you the impressions that I have gathered and see what you think of them by the end of this evening. We're getting new impressions of the sky. Once upon a time, clouds used to be puffy like this. Today, you see a lot of streaks, feathers, and unnatural crisscross configurations. When you look closely at what forms your impression, you can see that this is the impression version. You will see streaks, feathers, more feathers, lines. When you look closely at the lines, you see this. Looking at a collage of lines, with what appears to be a plane. You see ropes in the sky made by planes. Forming a web, a haze. The formation of man-made clouds is admitted to by NASA. They call it jet cirrus. We're told that new atmospheric conditions are causing jet contrails to linger in the sky. But anything that stays in the sky for many hours fans out, as you see here, and forms a layer of material thick enough to block sunlight for miles, is not condensation. The con in contrail is for condensation. So if it isn't condensation, meaning water droplets that under normal circumstances quickly evaporate, then what is it? Here's the result of one. It was a huge donut. This is Cardiff State Beach, where I live, and it literally formed a big oval, a halo in the sky. Contrails typically form behind high-flying jets, so that's high altitude, in low humidity. Clouds typically form at low altitude in higher humidity. So the rule is this: clouds and contrails need opposite conditions to form. But today, we have vapor trails, as they call them, condensation trails turning into clouds and spreading out to form a low-altitude white haze. In many parts of the world, we're losing our blue sky. You see New Mexico in the past and New Mexico in the present. The word for this, many people call it whiteout. If you travel in a plane, this is what you're going to see from the window. There's a demarcation line where the white arrow is. I call it the spray line, below which the world is seen through a veil, and above which you have that true deep blue sky. We live in the veil. We live in a white world of whatever it is that's descending on us from above. Our visibility is impaired. The sky, as we look up at it, is now a much lighter blue. But the change has been so incremental that most of us are too busy to have noticed. When you look at airborne environmental samples and the soil and water samples that people have collected throughout the country, you get three types of materials: metallic salts, filaments or fibers, and engineered biologicals. Metallic salts are oxides. Aluminum, titanium, barium are three examples. And it appears now that the air around us has been filled with these metallic oxides, which are conductive particles. So the air is no longer neutral. Air is supposed to be neutral to support life. It is not supposed to be charged. The air around us is also filled with fibers. And if you get a black light, apparently you can buy them at Home Depot, you can see, I've got to find my mouse, here it is, this. All right, how do you make clouds and rain? Clouds are formed from moisture that evaporates from the Earth's surface, carried upward on air that's warmed by the sun. As the warm air rises, it begins to cool. There's a certain point, they call it the dew point, D-E-W. And at that point, the moisture in the air begins to condense around particulates which are in the air. And these particulates are usually dust or salt. These particulates are called condensation nuclei. They have an affinity for moisture, dust, salt, like water, okay? As more water vapor gathers around the particulates, the combination becomes unstable, which is a raindrop. So we know that particulates in the atmosphere nucleate, meaning they gather into raindrops. Now here is something interesting. Daniel Rosenfeld, he's an atmospheric scientist, Hebrew University, Jerusalem. He claims that pollution particulates actually prevent rain by forming very tiny, very stable droplets that remain suspended and do not fall earthward. Now this was headlines. You can Google, pollution stops rain. If this is correct, then it suggests that the smaller the particulate, the more stable the droplet. So this is how you would get clouds to hang in the sky as haze, or so-called clouds. And so it would seem that you could create cloud-like masses simply by releasing small enough particulates into the atmosphere. And those clouds would not fall. They would spread outward instead to form haze. Which leads us to the subject of geoengineering, defined as the large-scale manipulation of the Earth's environment to suit human needs and promote habitability. Last year, scientists began to discuss something called SAG, stratospheric aerosol geoengineering, which was supposedly spraying soot and sulfates in the atmosphere to form a shield against the heat of the sun. This presumes the notion of man-made warming, the idea being that atmospheric science can rescue us from climate change, which is nature's retaliation against us for creating global warming. For now, we're told that this is a future consideration, but for years, we've already been seeing strange clouds and hazy skies. What exactly is dripping here? This is a photo I took, Cardiff, California, and I call these the bearded clouds. And here you have this new form, they tell us, new form of cloud, a hooked cloud called cirrus uncinus. And here you have asperatus. This is like the canopy effect, blanket. This is another picture I took. I had never seen this kind of cloud before. I'd only seen photos of it, and then it suddenly appeared. The presence of strange clouds, persistent haze, and impaired visibility is not just in the air and sky. It must be having an effect on the ground, on the earth's soil and water. Francis Mangles is a retired federal wildlife biologist and botanist from Mount Shasta. And he notes that between the 1950s and 60s, jets did not leave many contrails. Today, what appear to be jet contrails are much more frequent and last for 10 to 20 hours. And here's what they look like over these beautiful mountains and lakes. Now this would make a lovely view, don't you think? Counting all trails in every direction over Mount Shasta, Mangles says, the FAA indicates there can't be that many flights per day. The number has been as high as over 30 contrails at once, and I've counted 100 just before weather fronts. Typically, heavy spraying days are just before weather fronts that come in from the Pacific. So here you have a photo of this man who is a retired federal wildlife biologist and a water specialist. Lab tests from Northern California show very high levels of aluminum and barium in soil and pond water. Francis Mangles told me that at 100 micrograms per liter, when he was working for the federal government, you were shut down, 1,000 micrograms per liter. So I asked him what normal is, what's tolerable, and he said 5/10, 0.5 micrograms per liter is considered normal. So here you have the findings from pond water in Mount Shasta. You can see 12,000 micrograms per liter. UG is short for microgram, which is 24,000 times normal. Snowdrift at 8,000 feet, 61,100 micrograms per liter. Way, way past normal. And here you have a lined pond. So please picture a lined pond. A pond with a liner, vinyl something, in Bella Vista. And you got a reading, or the person who did this, I know him, Dane Wigington, got a reading 375,000 micrograms per liter in this little pond. Mangles has, I will repeat, a master's of science in water-related subjects. And he also reports that a house, the soil tested outside this house in Northern California was 3,000 times, 3,000 micrograms of aluminum more than the soil under the house. I'm sorry, I said 3,000 times. Anyway, so what does this mean? It means that something is dropping from above and is affecting the soil around the house, but not underneath it. So you can't say that this is in the groundwater. You can't say this is in the earth itself, coming from the earth. It's got to be coming from the environment. The dying of the trees. I've spoken to Charles Little. He's the author of this absolutely fantastic book, which you can buy still by going to half.com or Amazon used books. It is about the natural or biotic reasons for tree death, and also the effects of industry and deforestation, which began decades ago. When manufacturing exploded, acid rain, different forms of pollution started to affect trees. But what has come along since this book was written is this. Nowadays, we have an effect coming out of the environment that can't possibly be natural. Look at this corkscrew. I took this picture myself. This is the wine opener style. You have nature trying to cope with the introduction of new substances, particularly metallic oxides, which are also the metallic salts. Living things, as I said before, the metallic salts have an affinity for water. So living things take up these salts along with the water that they're seeking. The natural world, which gets its food from the ground, is sucking this stuff up. This is a scraggly pine tree outside the post office in Cardiff, California, where I go every day. And you can see how brittle it is, lots of needles lost. If you look closely at the needles when you stand under the tree, you see that they're all frosted with brown. Signs of tree death are these sagging limbs, these leafless, scraggly branches. You also see infestation by bugs, insects, mites, fungus. These are spider mites that have taken up in a tree that was right next to my house. The entire tree had to be felled because of these white webs in it that were killing off the tree. But again, I'll explain why in a minute. This isn't the primary source of the death. Here you have a phenomenon that Deborah Whitman, who's here with us today, has done a lot of research on. The white, we call it the tube sock effect on trees. You see white bark, and when we tested the bark of this tree, it showed it had these readings, aluminum 387. Now this is milligrams per kilogram because this is a mass analysis rather than a liquid context. So you have barium at 18.4, strontium 113, and titanium 15.2. You can see again in these photos the enormous scorched look. That's a very dying, very unhealthy tree. And you see this other thing called secondary growth. The tufts on the trunk poking out, that's the tree giving itself, desperately trying to give itself a second stab at life. So when you look around you and you see these little growths, sometimes they're all the way along the trunk from the ground up. The beauty of an induced problem is that its twin is the solution. As nature begins to die, we will be told that science will have to step in to save it. So, you'll have GMO trees as the answer. The silent forest, they call it, genetically engineered trees, non-reproductive, no fruit, nuts, blossoms, no insects, animals, birds. Low lignin, that's the wood fiber, which makes the tree very easy to cut and pulp. The silent forest will grow straight and tall and will be replenished by the state in what will be considered appropriate numbers and in appropriate locations. In May 2010, from this article, we learned that the USDA approved large-scale field trials of 200,000 GMO eucalyptus trees made by ArborGen, biotech company, to be planted from Florida to Texas. Now, we're told that the purpose of the trial planting is to evaluate whether such GM trees can become new sources of wood for paper and biofuels, also in the name of conservation and improvement. This is the story they give us. We're trying to conserve, we're trying to go green, we're trying to help. But then you come up against articles like this, 2008. This was from something called the MIT Technology Review, an article announcing biochemistry's development of toxicity-resistant crops. In particular, in this article, aluminum-resistant plants. So the article tells us that aluminum in soil stunts the growth of crops. Wheat, corn, and barley don't fare well in aluminum-laden soils. But now, scientists, plant scientists, have found a way to get the plant to shut down its own cell division. To stop the plant from shutting down its own cell division. Because when a plant encounters toxins in the soil, it says to itself, I don't want to keep growing. So it shuts down the cell division. But they have figured out a way to keep prompting those plants to produce reproduction. So this is a gene mutation, a single gene mutation that inactivates a protein so that the plant continues to grow. The quote from this article is very interesting. The plant is effectively blind to what's happening in the cell. And that's from biochemist Paul Larson. The mutant plants can maintain high levels of growth in the presence of toxic levels of aluminum, even if they sustain DNA damage. When something begins to die in nature, it attracts bugs, blight, molds, even viruses and bacteria. This is nature's way of hastening decomposition so that the dying form can become food for other living things. Today, we have an epidemic of tree decline all over the world, not just in America, but it's in Australia, it's in Europe. Thousands of square miles of die-off in the savannas and forests of many, many continents. And in cities and suburbs, trees are rotting as they stand, swooning, breaking. They are hazards to property, requiring removal. 6 trees around my house have been taken out in the last 2 years. Sunlight is a natural disinfectant. As hazy skies limit the sunlight, molds and fungus grow. As plants that are taking up toxins struggle to live, molds, viruses, bacteria begin to take them over. This is all part of nature, and bioremediation will be the obvious answer. Metallic salts have made our air conductive. This means that we and everything around us can transmit and propagate energy. The air is no longer neutral. It no longer supports, in a healthy way, living things. The second group of materials found in these environmental samples is unidentifiable fibers. And I really want you to appreciate the meaning of unidentifiable. These fibers have been sent to sophisticated laboratories, and there is nothing in the databases that match them. So these are fibers, we could say, that do not exist in nature. People around the world are developing lesions on their skin that ooze and produce fibers. This is known as Morgellons syndrome. Tissue samples cultured from ordinary people without this ailment contain and grow the very same fibers. Here's a 5-inch lesion on a woman's body that has never healed, 5 inches. The fibers or filaments are actually tubules with hollow insides. When these fibers are cultured, they produce colonies of filaments. You can see the extension filaments. And these colonies continue to grow and reproduce, branching out into more filaments and more colonies. The filament cultures can be grown from saliva samples, tissue from the skin, mucus, urine, blood, from animals and people, regardless of the presence of the Morgellons condition. The fibers are segmented with visible structures inside them. So where do these fibers or filaments come from? Airborne environmental samples that were collected by Clifford Carnicom, he is a researcher in this subject, in 2000, the year 2000, gathered at high altitude on a mountain in New Mexico, showed the presence of those fibers whose structure matches exactly the tubular filaments. When I say fibers and filaments, basically interchangeable, showing up in our blood, tissues, and skin. Additionally, the samples collected by Mr. Carnicom showed what he calls, and was called in biology, desiccated erythrocytes. This is a multisyllabic term, but it means dried red blood cells. So, why these were in the air? Why are dried red blood cells in the air? Very puzzling. A medical microscopist, a biologist specializing in microscopy, confirmed to him that they were human red blood cells, but that they had been engineered in some way so as to be preserved. So again, you have to ask yourself, if I were 5 years old, I would say, Mommy, what are red blood cells doing falling out of the sky? Biology divides life into 3 kingdoms: bacteria, archaea, and eukarya. Bacteria and archaea are simple organisms with no subcellular compartments. Eukarya are complex with defined cell compartments and internal organelles, mini organs like mitochondria that make DNA and energy for the cell. Plants, animals, and humans are eukaryotic, as are fungi and slime molds. The archaea are the hardiest of the life forms. They're able to withstand grinding pressure, heat, acidity, and alkalinity. They can live in volcanoes, geysers, and the ice shelf. Bacteria, on the other hand, will die in extreme heat and cold, which is why we cook and freeze our food. Now, the materials appearing within these filaments are as tough as archaea. They look like bacteria, and they self-replicate. One looks like something called mycoplasma, but is not. One looks like chlamydia, but is not. And natural fibers or filaments would be classified as fungal in the domain of eukaryotes. But these fibers contain forms from the other 2 groups inside them. You're looking at a tubular fiber with self-replicating internal elements resembling bacteria and behaving like archaea. That does not happen in the natural world. So what are they? What's going on? It appears that within our bodies we now have all 3 life kingdoms replicating themselves. What does that make us? Are we still eukaryotes, or are we becoming something else? You could call this transbiology, a crossing of biology, the creation of hybrid forms. Materials are forming in our bodies that are not native to us, not natural, and entirely new. You can see in these pictures the tubular filaments with the branches. They're branching off. That's their self-replication. They're creating new colonies. And you can also see in the bottom picture a little bud. That's the beginning of a new fiber. The territory of things on the scale of atoms and molecules is the nano world. Science has opened up the nano universe, where incredible new creations are possible. Nanotechnology explores materials that are less than a micron in size, or from 1 to 100 nanometers. And a nanometer is a billionth, 1/1000th of a meter. Nature and biology continually work on the nanoscale, assembling proteins and building with crystals to conduct the business of life. Spider webs are an example of that. Very fine filaments with enormous strength and flexibility because they contain nanoscale crystals. So let's go back to the filaments and the structures they contain, these ones that Clifford Carnicom has found. In those unidentifiable filaments, he has observed the formation of red blood cells and submicron-sized structures. So now you've got a filament making its own red blood cells. The engineered cells, the red blood, very, very tough, withstanding excesses of heat and chemicals, indicating that they are designed to endure almost anything. He has put them in a Bunsen flame, he has poured bleach on them, acid, and they still endure. In addition, they are able to replicate, growing outside of the body in a petri dish. This is highly sophisticated technology going on by itself, not in a laboratory. So we're witnessing red blood growing outside, having nothing to do with the bone marrow, in a petri dish, in somebody's very low-level lab. Could it be that artificial materials are being introduced into living things? Here you have, this comes from the nanotech industry. It's what I call the nanotech pyramid. You start with materials, you go up to structures, processes, and devices. Could it be that artificial materials are being introduced into us to create new processes from the ground up, which processes will override our own natural biology, our own internal systems? People with advanced Morgellons syndrome started with fibers coming out of their skin, and they're now observing very strange crystalline forms and metallic devices. The fibers became multicolored, and now they were accompanied by plaques, strange, hard little, almost like shards. People say they're like glass, and they're colored. The lesions keep emitting these things. They don't heal over or close, sometimes for years. Here are more plaque structures, even grooved metallic devices. On the right, you'll see one device, the front and the back. These images are supplied by Jan Smith of MorgellonsExposed.com, who has done extensive research on biotech websites, looking for things that match what comes out of her body, and she's finding them. The fibers from her skin were from high-density polyethylene. And I'm going to read what she's written on her website. The bizarre nature of my findings suggests a man-made source. It occurred to me that if these pathogens were being bioengineered in a lab, they were made of multiple components. The mutated material might reproduce and intermittently send out a batch of identifiable debris, much like the original genetics. I call this type of debris throwbacks. It's her theory that when new life forms are made through this gene splicing and mutation, bioengineering and nanotechnology, they will self-replicate in all kinds of ways, creating different mutations in each generation. Morgellons specimens can sometimes be identified with the original spores before those mutations occurred. Jan found 3 varieties of something called Omycota fungus in her specimens, as well as something else called Dictyostelium discoideum, which is a slime mold that is a major player in biomedical research today. The material coming from her body was ribbony and motile. It was grooved and oval, very much like the Omycota spore you see here. At the bottom, where the purple arrow from the bottom is it's a diagram of Omycota. And on the upper right, you see something that came out of Jan's body that looks exactly like Omycota. The fiber strands that you see here were made of cellulose and GNA. GNA is glucans polysaccharide sugars, and this was confirmed by microscopy. GNA is a synthetic cousin of DNA. It has a 3-carbon glycerol sugar in it instead of the 5-carbon that you find in DNA. Here's a news report from April 2008 in which we are told, big headlines, GNA is DNA's chemical cousin. It's a nanotechnology building block. The Biodesign Institute at Arizona State develops the first self-assembled nanostructures composed entirely of GNA, synthetic analog of DNA. The nanostructures contain additional properties not found in DNA, including the ability to form mirror image structures. An NIH article from Japan, on research from Japan, also tells us that various conductive polymers and gold nanoparticles are entrapped within the helical superstructure of this GNA. Now, the particular sugar found in this GNA is called beta-1,3-glucans. And here you see it under what's called a Raman spectroscope, very sophisticated equipment. It looks, the microscopist who looked at this called it a sugar snake. So this is the GNA that you're looking at. When Jan's fibers were put to a high heat flame, they released what she calls a gold payload. So they dropped out this tiny little bead of gold. And here's more. The fibers that contain a head, she calls it a golden head, orbs, and they all have this payload in them. Here you have a nano array that she found, a couple of them in her body. Nano array is a tiny device used in biotech for DNA hybridization. And on the left, you see a diagram from an industry website of a nano array. And on the slide, you can see actually two of them. She's marked them with red X's that came out of her body, looking exactly like this. And again, I'll say it again. The nano arrays are these tiny devices that are used for DNA hybridization. So, I'm going to repeat my nanotech pyramid. We begin with the emergence of basic filaments, followed by more complex structures. So what processes are going on here? Are these materials combining to form devices that are working together inside us? What is happening to our biology? And remember that tissue samples obtained from ordinary people who have no Morgellons symptoms, no lesions, can be cultured to produce the very same filaments found in people with Morgellons. So we would have to conclude that Morgellons is like the canary in the coal mine, and that only some people are exuding the materials. And could that be because their bodies are rejecting it, while our bodies are integrating it? Jan's body has been sending out these plaques, colored plaques, which are hard pieces of silica. Some of them have these dots on them. They're very small. They require handling with a needle as she places them under a microscope. The plaques are fragile. They can shatter.